So, welcome back to our next session. As you see, we will do it in English. And also the topic of aging might be in the far future for our main target group at the European Researchers' Night. It is a matter of us all. More than that, it is a worldwide concern. Therefore, the European Union funded a doctoral program called Hydra. Young PhD students from all over the world have been brought to Innsbruck to do research dealing with aspects of aging, drugs, and regenerations. And it's a pleasure for us that 4 of them are here today to give us insights in 3 fields of their astonishing research. First of all, I want to welcome Camille Brucker. She will talk about aging research. So welcome, Camille. And the stage is yours.
Super. Thanks, Julia, for this introduction, this nice introduction. So hello, everybody. And so I will start to present you concerning the aging research. So first of all, I will start with some key facts from the World Health Organization. So what you should know is that population aging is a global phenomenon, and it's occurring all around the world. Some idea of numbers of global average life expectancy. You should know that in 1950, the life expectancy was around 46 years old and 67 years old in Austria, which is already quite nice at this time. And almost 70 years after, in 2019, this global average life expectancy rise to 70 years old and 81 years old in Austria. So it's a huge increase, and this was possible thanks to public health access and also all the medical advancement we have done since this last decades. So you can get older and be enjoying your old age also and keep going to dance, if I can say. And to give you also some other numbers, in 2015 there were around 900 million of people aged 60 years and more, which was around 12% of the total worldwide population. And the expectation in 2050 are 2 billion of people aged of 60 years and more, which will represent 22% of the total worldwide population, and means that 1 person on 5 will be aged 60 years and more. So on one side, this is very, very nice that we are getting older and older. But on the other side, also there's a declining in the fertility. And the fact there's also an increasing longevity leads to rising numbers of older persons. And so this already happened in 2019 from the BBC in an article saying that there are now around 705 million people over 65 on the planet, while those aged 0 to 4 numbers about 680 million. So this will represent a challenge for the society, especially for the healthcare system, also the social system. But this is not the part that we are interested in, of course. It's more about the biological and the research. So what is aging exactly? So from Wikipedia, the definition of aging is the process of becoming older, which is very explicit and informative. And here are some pictures of what people imagine of aging. And what you should know is that aging starts quite early in our life, around 25 years old. So when we are children, teenage, we are growing up until young adults. And once this growth phase, if I can say, is done, we start to age, unfortunately. And what we see usually from aging at the beginning, it's visible or marks. So if I can say from the outer shell, so it's the appearance of wrinkles and loss of skin elasticity. And so people are using a lot of anti-aging cream and also doing some hyaluronic injection to avoid this kind of wrinkles and this loss of skin elasticity. But you are just treating a symptom, if I can say, the consequence of aging and not the first reason, which is aging. And besides this, you have also a progressive loss of hearing and sight, and also back and neck pain and osteoarthritis. So on one side, it seems not so funny to age, unfortunately. But this is only the tip of the iceberg, if I can say, and that all people notice. And me, I'm more interested in what is going on under this, what is hiding, if I can say. And from a scientific point of view, aging can be defined as a time-related deterioration of the physiological function necessary for survival and fertility. In other words, that means that aging process slowly leads to a decline in the function of our organs. So the inner shell, if I can say, what is inside our body. And so this leads to a gradual decrease in physical and mental capacities. And also the reason what we are studying aging and investigating this process is because more you get older, more you have the risk of having apparition of age-related disease, especially type 2 diabetes, cancer, cardiovascular disease, and also neurodegenerative disease as dementia, Alzheimer's disease, or Parkinson's disease. And something you should know also is not depending, aging process depends on everybody. It's specific to each individual. Maybe you already saw some old people of more than 70 years old, 80 years old, doing a lot of sport, enjoying their life and being totally independent, and some other people of the same age who are not able to move anymore or totally dependent on the other people. So aging is very specific to each individual, and you should know that your lifestyle or what you are doing during your life influence the way you are aging. And there's three main factors that influence our way of aging, which are genetics, individual lifestyle, and external environmental influence. From the genetic part, unfortunately, you cannot control it. You just have to hope that you have a good genetic heritage. So from your family, your parents, but from the individual lifestyle and also the external environmental influence, you can control these factors. So I will give you just a few examples of it. So from the individual lifestyle, this can be controlled from the food, so what you are eating, and also from the physical activity. And from the external environmental influence, it's more related to the stress. So from the school, the study, the work, also your social life, also with all the social media, now I'm thinking all the anxiety it can generate for the young people, but also from the environment with neighborhood, in your family, where you are living also, if it's in a town, in a village, also with the pollution. So all these factors impact the way that you are aging. And I will just give you a clear example, because I'm working more on this aspect with food, which is about dietary restriction. So maybe you already heard of it. And it's corresponds to the fact that you are eating a little less than what your body needs in terms of calories. And it has been shown that this lifestyle, if I can say, performed without malnutrition is the most robust anti-aging intervention known so far. And it has been shown since decades that it extends healthy lifespan in a wide variety of species, from the yeast to the human, passing by worms, also Drosophila, so which is a fly, mice, fish, and monkeys. And I'm interested in this aspect because it had been shown that weight loss intervention can extend lifespan of obese people. So you should know that obesity is a public health issue worldwide, all around the world, and it's defined by the body mass index. Then this one is superior to 30, that means the person is obese, and it has been observed that in people who lose weight and gain normal weight, they observe that there is a reduction of the fat mass and a reprogramming of adipose tissue function, and this postpones age-related disease, because it has been shown that obesity is linked with age-related disease, because you can have type 2 diabetes, also cardiovascular disease, and obesity is excess of fat, white fat in the body, and this leads to a dysfunctional adipose tissue. Adipose tissue is a scientific name for fat, if I can say. And so after weight loss intervention in these obese people, so by bariatric surgery and or long-term dietary restriction, we observe there's a reprogramming on the adipose tissue function, And you should know that adipose tissue is not only a storage organ, that's what everybody thinks because it's just fat, but it's actually an endocrine organ which is interacting with the whole body, especially with the brain, the intestine, and also the heart. So if your fat is doing well, the rest of your body is doing well. Too much fat is not good, not enough fat is not good too, so you need a certain balance. And in my lab, so in Innsbruck, we have the chance to work with adipose tissue for formerly obese people. And on this, we study it, and we try to understand this reprogramming happening after this weight loss, and try to understand the mechanism on how weight loss improves health span in this individual. So this is one strategy I show you, that we investigate the aging process itself, but there's also some other strategy that my friends will present to you after, which is the regeneration capacity of some very curious animals, if I can say, that we can try to implement it in a human and be able to regenerate ourselves and be again like teenage, maybe would be nice. And you have also another part which is very important in every kind of research, which is drug research. And so, of course, finding the way to slow down aging, stop it, or be able to regenerate will be like finding the holy grail. And in all this, the wish is not to, all this research is not done in the wish to age longer, but healthier, because it's better to live longer in a good shape than longer in a bad shape, of course. And yeah, to finish, you should not be afraid of getting older. You know, age is just a number and is not defining the way you are aging. So yeah, thank you for your attention.
So thank you, Camille. So that sounds good. Okay, it's only a number. I will tell me this when I look into the mirror every day.
You know, in France also we say with the wine, you know, more it gets older, better is in taste also. So you know, with time we get better and better. That's what we say.
Okay, yeah, that's true. That's true. So great. Thank you for the insights into your research. So let's take a look at the chat and the questions. And yes, what I would like to ask you is, do you think that a cure against aging will be found, the holy grail? Will this be possible in the far future? So we take some pills and we stay to 35?
That would be nice. I think this will be possible. On one side, we are able to do a lot of things nowadays, which are quite impressive. We are already thinking that we can go on Mars. So I think we are able to find something for sure to slow down aging, stop it. We will see. But aging is quite very complex process. It's not only one mechanism, it's different mechanisms all together. So maybe stop totally aging, maybe one day it will be possible, but if you have to take 20 pills for that, it will be a little weird. But maybe to slow down for sure, maybe we'll be able to live until 120 years old and be able still to run a marathon, for example, or move everywhere without any trouble. I think it's possible.
Okay. Is it sometimes frustrating? So another question in the chat. So is it sometimes frustrating to work in the field of aging? What motivates you in your work?
It's not frustrating. I'm not afraid to get older. This field is very interesting because at the beginning I was interested by adipose tissue itself and all metabolism. The body is very interesting machine, if I can say. And the way that we are able to metabolize what we are eating, that's why I was very interested in it. And as obesity is a public health issue, and as it's linked with aging because it accelerates aging, that's why I was interested to work in this aspect. And also the fact that we are working with adipose tissue from human, that's also a plus because it's more, it's directly to human and not passing by animals. So it's more realistic, if I can say. And yeah, so that's why it motivated me to work in this field.
Okay, so great. So we have 1 minute left. So one last question. So from the chat, so some scientists who the poster knows recommend dinner canceling 1 to 2 times per week. What do you think of this dinner canceling?
Yeah, it's, I mean, it's one in category of intermittent fasting, if I can say. Yeah, there's a lot of study which have been done on this. And I mean, what I think personally, I already done intermittent fasting, if I can say, quitting the breakfast. And I mean, I didn't feel so good. I mean, it depends of people. For some people, it has benefits. For some others, not, because it creates frustration. And after they're eating a lot or not good things. So, I mean, it's not useless, but I mean, yeah, there's a lot of publication saying that it's benefits for you. Also doing sometimes total fasting, but I mean, it's not, yeah, it depends of people. I mean, you have to, because sometimes it's complicated to fit this in your lifestyle also with the work or the things you have to do besides. So sometimes it's complicated to implement these kind of things. But from my personal point of view, I mean, people can try, and if they like it, why not? Because at the end, it's beneficial. I mean, yeah, it brings benefits from the literature.
Okay. So thank you very much, Camille.
Yeah, thank you.
Thank you. So next, I would like to welcome Marion Lechable, I hope I spell it right, and Alessandro Pennati, who will, beside others, tell us how the well-known monster Hydra from the Greek mythology might help us to regenerate faster after injuries, or what we can learn from Hydra. So a very warm welcome to you, Alessandra and Marion. And now it's your turn.
Okay, thank you. So, okay. Can you see the pointer?
Yes, we see it.
Okay, thank you. So, good evening, everybody. Thank you very much for the introduction. As Julia already said, we are two PhD students from the University of Innsbruck, and today we are going to talk about regeneration research, and we will show you some models that we use for regeneration studies in our university. So here you can see pictures of different animals that share something in common. In fact, all these animals are able to do a process called regeneration. But what is exactly regeneration? As many of you probably know, lizards are able to regrow their tail after losing it. And this is only one example of regeneration in the animal kingdom. But different animals can have several types of regeneration process. So indeed, regeneration can be applied to cells, tissues, organs, or also to the whole body. For example, cells from skin, muscles, or bone can regenerate in humans, so we can regrow injured tissues, while the axolotl can regenerate almost all his organs within some weeks. The sea stars, on the other hand, is able to regrow the entire body, even from a tiny piece. So why do we study regeneration? The first reason is to understand the principle of this mechanism, and why only some animals are able to do extensive regeneration while other animals, like humans, have only limited regeneration capacity that can be extended only to some cells or tissues. And the second purpose is to study regeneration for its potential uses in medicines, for example, to treat a variety of injuries or diseases. And this field of research is called regenerative medicine. And with this research, we hope to learn more about the human aging process. But now you might ask yourself, how is regeneration related to aging? And in general, organism and human can regenerate some of their cells when needed during the lifespan. However, this ability gradually declines over time. But this is not the case for some other animals. which are known to not lose this capacity, because they are not aging and seems to be immortal, like, for example, this little jellyfish. So in order to better understand how these changes in the regenerative ability occurs during aging, scientists and also us are studying these animals that show only few signs of aging during their life. And there are different model organisms that can be used to study this process. And we are going to present 3 of them that we are using in our university. So here you see pictures of 3 models that we are using. So first we will talk about the freshwater polyp Hydra, then the sea squirt tunicate, and finally the tropical fish, zebrafish. So you probably know Hydra from the Greek mythology, which is represented as the many-headed monster called the Lernaean Hydra. And in the myth, this monster is able to regrow his head after being cut, which makes this animal immortal. But in reality, Hydra is just a tiny freshwater polyp, but it is immortal also in real life. This animal is part of the same group of jellyfish and corals, even if they don't, even if they look very different. The interesting thing about this animal is that they possess extensive regenerative ability that can be applied from a single cell to the whole body. Indeed, as you can see from this video, when a Hydra is cut in 2 pieces, the part with the head will regenerate a foot, and the part with the foot will regenerate a head. And in the end, what we will have will be 2 new Hydra. So the second model we are studying is commonly called the sea squirt. They live fixed to the substrate, to the sea bottom, sorry, and they filter the seawater to get food, as you can see from the video on the right. They are used for developmental and ecotoxicological studies, and recently they arose as a regeneration model. From this picture, you would think that the sea squirt tunicates on the left resemble very much the sponges on the right. But in reality, they are really different animals and very distant in the evolution. What is really exciting about this animal is that they are really closely related to vertebrates like human, like us. So studying these animals allow us to understand some basal mechanisms that evolved and gave rise to the one of humans. There are different type of tunicates. Some lives in colonies, like the one that you can see in this video, and other are solitary animals. The colonial one, like this one, present extensive regeneration capacity, while the solitarian one are more limited to regrow only just part of their body. So the last model that we use is zebrafish, which is a tropical fish and is very popular in the aquariums. So you might have seen already this animal in pet shops. It's one of the most common and studied model organisms within the vertebrates after mouse. And they are widely used in medical research and drug development because of their close relationship with humans. Indeed, they possess similar organs and present similar disease like Parkinson or diabetes. Zebrafish is able to regenerate almost all its organs, even during the adult life. And one advantage of using zebrafish is that in 2 days, all their organs are already formed. And thanks to the transparency of their body, we can easily see all the organs and mark them to follow the regeneration process. Another advantage is that they are really easy to manipulate during the first stages of development, and mutation to the DNA can be easily performed in order to study the gene function. So now you might ask yourself why these three animals can do regeneration, but we cannot. And at this point, I will let my colleague Marion explain you more in detail.
So thank you, Alessandro. So we know that most animals, including humans, cannot regenerate. In fact, during the first week of development of human embryo, they possess special cells called embryonic stem cells that are the first progenitors of all the cells that compose our body. These cells divide and create a new pool of cells called adult stem cells. And these cells are able to produce only a single type of specialized cells. So for example, these adult stem cells can only give rise to neurons, or this one to skin cells. In the end, only the adult stem cells remain during our entire life, but in a limited number with limited functions. On the other hand, in the case of animals with extensive regeneration ability like Hydra, the situation is a little bit different. So we know that stem cells of Hydra can, of course, give rise to any type of cells like in human, but they can also give rise to more stem cells that will be, again, able to divide into different cells. And this is possible over their entire lifespan, which is not the case in human. The fact that Hydra keep these stem cells make them able to regenerate during their entire life without any decline over time. So as we said before, humans don't possess embryonic stem cells during their adult life. However, in 2012, Shinya Yamanaka discovered a way to obtain cells that possess almost all the characteristics of embryonic stem cells from specialized cells. Indeed, he found that the induction of 4 genes called the Yamanaka factors are sufficient to reprogram any type of cells into a stem cell. And this discovery has been awarded with a Nobel Prize, and this technique is now worldwide used to reprogram the cell fate. For example, nowadays we are able to take cells from our skin and to induce them into stem cells with these Yamanaka factors. And then these induced stem cells can become another type of cells like neurons. But even more fascinating, we are able to grow a prototype of new organs that can be used for research purposes, and this is called organoids. So here you can see a picture of tiny brain organoids grown in the lab. And on the bottom, you can see a time lapse of their development under the microscope. So these organoids are a really innovative technique that will be used more and more in the future. And researchers have the hope that by using brain organoids as a model, for example, we will be able to understand better how the brain works and how diseases like Alzheimer's are affecting the brain. So in conclusion, today we showed you an overview of what regeneration is, but in reality, we are far from understanding the precise mechanisms behind it. We know that stem cells might be the key player in the regeneration. However, many questions remain open. For example, what regulates these cells and which genes are involved in the process remains unknown. And in addition to that, we are not sure that the stem cells are the only player in the context of regeneration, and what trigger exactly the initial response of regeneration after injuries is unknown. For this reason, everyone involved in this field of research is focusing on one piece of the puzzle, and with the hope that one day we will be able to assemble all the pieces and have a complete picture of what's going on. So to finish our talk, we would like to explain you briefly what we are doing in our university. So because regeneration is a huge field of research, and the 4 of us, we are focusing on special aspects of regeneration. So for instance, Narges is characterizing stem cells in zebrafish pancreas. Alessandro is trying to decipher regulation of stem cells related genes in tunicates development. Angelique is working on neural regeneration using induced stem cells. And on my side, I'm working on stem cell mechanisms during Hydra regeneration. So we would like to thank you for your attention, and we are happy to answer questions.
So thank you, Alessandro and Marion. Thank you very much for your presentation. So when so many people are working on this, maybe sometimes in the far future, I can cut off my finger and it grows then again. Would this be the target?
Maybe.
Maybe. Okay.
I mean, we are really far from this, but
but maybe. Okay.
Yeah,
so
who knows?
I have to get old. So yeah, I have to follow the advices of your colleague before. So then maybe I can do this. Yes. Thank you very much for your presentation. And as we are already in time, so we are still in time, but we somehow don't have really time for questions. I would like to go further to Ferenc Török. So last but not least, I would like to welcome Ferenc Török, who will take us on an exciting journey through time in drug development. And Ferenc, are you here?
Ferenc, yes.
Hello. Did I spell it right?
No, Ferenc.
No.
Okay,
sorry.
At the end.
Okay, so sorry.
It's fine. Do you hear me?
Yes, we hear you. We see you. So now it's your turn. Take us into a time journey.
Do you see my presentation as well? Is it on? Hello?
Yes, we see the screen, but we don't see your picture. If you have a webcam, you could turn it on.
This is on.
Okay.
I'm sharing the full screen, so I don't know what can. Just a second.
But we see the presentation.
It's okay. Yes.
Yeah,
So thank you very much for inviting for this presentation. My name is Ferenc Török, and I'm one of the 5 people doing drug research in the frame of this PhD program. Currently, I'm working at the Department of Pharmacology and Toxicology at the University of Innsbruck. And to be honest, I struggled a bit when I prepared for this presentation because I wanted to show that how the drug research started for the humanity, but I found no exact time point because the more I digged in, I realized that this is not even a human thing to aim for health. It's enough just to think on the house cats eating grass to regurgitate the swollen hair, or thinking on ants cultivating different fungus species to keep the colony bacterium-free, undermining the aseptic and antibiotic effect of this fungus. But coming from here to this point that you can see on the bottom picture is extremely long and bumpy road. We just have to think on, for example, the Stone Age people using crude herbs and plants and fungus to undermine also the benefits as drugs, or ancient Egyptian people using different remedies for mummification. As well, they were the first people who made anatomy atlases as well, and did certain kind of scientific approach during their work. In the Greek and Roman times, it was also, considering the lack of gadgets and different tools, people could only use the crude compounds directly obtained from different animals and plants, such as the venom of the meadow viper that you can see in the middle and in my hand, a big female. That venom was used to cure different wounds, as you can see here, from bottles to where the blood clotting effect of the venom was undermined. In the medieval times, there was a certain approach that was lacking the scientific way of thinking. First of all, different health issues were supposed to come from God, and not from the misbehaving of the human body. So the most common thing for medication was spraying, and in addition there were some very primitive methods such as the vein cutting and the leeches in order to remain the so-called bad blood from the human body, or, which is a bit curious, the trepanning, where the human skull was opened in order to eliminate migraine, or to do very primitive brain surgery after battle wounds. That was the first time as well when the first pandemic occurred in the human history, wiping out approximately half the population of Europe. And in addition, there was a decline in hygiene as well, because the only place where people could receive hot water for bath was the public baths, but they were shut down by the church, considering their blasphemic background. In this era, basically the alchemists were the only people who used crude scientific approach during their work. They didn't have that much to do with healthcare or drug research. Their aim was to turn some basic metals such as aluminium, copper, and zinc into noble metals such as gold. So they invented a lot of methodology that were used later on in drug research as well, such as tools that are used in the laboratory or purification steps. When the scientific approach was set back on track was the Renaissance age, as you can see with Leonardo da Vinci. The Industrial Revolution also had its effect on the drug research. In the middle you can see Friedrich Jakob Merck, who was the first one who invented a scientific drug company approximately 350 years ago that is still working and still running. Antony von Leeuwenhoek was the guy who invented the microscope. So afterwards, in the 17th century, it was known that not the water itself is the one that is causing problems for health, but the microorganisms were discovered. And Edward Jenner at the end of the 18th century was the one who was the godfather of the vaccination. In the 19th century, Louis Pasteur, Emil Fischer, and Friedrich Bayer were the ones who had the biggest input on the drug research. Louis Pasteur with the epidemiology and the Pasteur Institute itself with the developed vaccination. Friedrich Bayer with the Bayer pharmaceutical company, and Emil Fischer was the godfather of organic chemistry. Despite its huge development, it was very far from perfect. As you can see, some drugs from that era, such as crude tobacco used as asthma cigarettes, the one-night cough syrup that gave alcohol, cannabis, and morphine together against coughing, or this, let's say, sleeping drops containing alcohol and opium together. The 20th century was a huge boost for drug research and life science researchers as well. Just have to think on Alexander Fleming with the antibiotics, or Jansen-Dürr and Krieg, first showing the human DNA structure, using the methodology that are still currently used in the laboratory nowadays as well. For example, the HPLC that is used for separation of different molecules, or NMR and X-ray crystallography to determine their three-dimensional structures. But where are we going right now? On this picture, you can see my colleague Ludovica in front of the computer, and these here are not confettis. These are different molecular structures on her computer, and she is simulating drug and protein interactions in order to eliminate long-lasting animal and laboratory experiments, in order to speed down our research. At the current stage, this is not a standalone methodology, but in order to make preliminary screening for drugs, which can be used later on, is a very good method. For example, in her work, she performed in silico analysis with approximately 1,500,000 different molecules, And afterwards, she came up with 12 compounds that will be tested afterwards. So she made a really, really huge amount of work. Besides the in silico modeling, I would like to show some examples as well that are now can be used in the future as well, and providing really robust research potencies, such as monoclonal antibodies. These are designed antibodies that are used against specific targets, for example, breast cancer or autoimmune disease. The drug targeting, you can see here the buckyball, which is a carbon-based drug delivering apparatus, being responsible to carry the drug itself in the human body to the proper places. And we shouldn't forget the gene technology, which is also the future of our research methods. But what are we exactly doing? On this slide, you can see the 5 people doing drug research in the frame of the PhD program. First of all, let me introduce Eduardo from Mexico. He's working with lichen, and after the dry freezing of the compounds, he tries to separate different compounds and test them in vitro and in vivo as well, in order to look for some promising bioactive molecules that can be later on used for drug research as well. But what happens if we find something? Here comes Aldo's role, who is currently working with different compounds obtained from coral species. Here in the laboratory, he tries to synthesize them in order to eliminate the need for harvesting of these species. Normally, these compounds can be found in these creatures in a very low amount, and this is much easier if they can be synthesized. Lucas from Germany is working with a molecule called rapamycin. This is also related to the previous presentation, because it is known that the rapamycin molecule can prolong life, and that has been shown with the flatworms, with flies, and with mice as well. And currently he is testing it on human tissues. And beside that, there's the dog aging project, where rapamycin is given for dogs in order to see if it can prolong the life of dogs or not, maybe. And here you can see Xu Chen, a Chinese lady, who is doing molecular modeling on a computer. From these amino acid sequences, she builds up the three-dimensional models, and afterwards she tests different drugs in order to see if they bind or not, or can be used for that purposes or not. But how does this work exactly? And here comes my role, because I've worked with voltage-gated calcium channels. These are proteins that can be found in different electrically excitable cells, such as nerve cells, muscle cells, pancreatic cells, and so on and so on. And these are responsible for the trafficking of the calcium ion, initiating action potential, muscle contraction, hormone release, and so on and so on. But unfortunately, these ion channels can dysfunction as well, and they can cause really severe problems, such as Parkinson's disease, migraine, epilepsy, autism, and so on and so on. And my task is to identify various compounds from various animals that you can see here on the left-hand side, to test them on these ion channels if they can be modified with these toxins. Normally, these toxins block the ion channels of the prey items in order to slow them down or inhibit the breathtaking. But after some modification, they can be used as drugs as well. For example, the zaconotide is from the cone snail, from the Australian cone snail species. Currently, this is in use as a very severe painkiller, because in comparison with the opioids, it has approximately 1,000 times higher affinity towards these ion channels, and in addition, it doesn't cause addiction as well. I would like to say thank you for everyone helping to set up this presentation, primarily the other doctoral research members and Monica Gerbusing for the administrative support. And I would like to say thank you for everyone who was listening to me.
Ferenc, thank you very much.
Yes.
Ferenc, yes, thank you very much for this interesting insights into your research. And a big thank you from my side to all of you in this program doing research on such an important topic.